Hey everyone, and welcome. Today, we're going to dive into something absolutely fundamental in pathology, the story of a single cell under stress. We're going to follow its journey from being perfectly happy and healthy all the way to its breaking point. Trust me, understanding this process is the key to understanding, well, pretty much all of disease. So here's our roadmap for today. We'll start with what it actually means for a cell to be injured. Then we'll look at the first critical stage, reversible injury. After that, we'll talk about the most dramatic part, the point of no return. Then comes stage 2, irreversible injury. And finally, we'll wrap it all up by connecting it to what you'll actually see in the clinic. Ready? Let's get into it. So first things first, what even is cell injury? Well, in the simplest terms, it's when a cell gets hit with some kind of stress and just can't keep its internal environment stable anymore. It can't maintain homeostasis. I want you to think of a cell like a rubber band. You can stretch it, you can pull on it, and as long as you don't go too far, it'll snap right back. That's reversible injury. But if you pull too hard, it breaks, and that, my friends, is irreversible injury. Okay, so let's start with that first phase where our cell is definitely feeling the strain, but it hasn't broken yet. This is reversible injury. The big idea here is that there's still hope. If we can just get in there and remove whatever's causing the stress, the cell has this amazing ability to fix itself and get back to normal. So how does this whole thing kick off? It usually starts with an energy crisis. Imagine a stressor like ischemia, a lack of blood flow. That means less oxygen, which means the cell can't make enough ATP. Now, ATP is the cell's energy cache, and without it, critical machinery starts to fail. The first thing to go, the sodium-potassium pumps in the cell membrane. When those pumps stop working, sodium rushes into the cell. And you know what always follows sodium? Water. So water floods in, and the cell starts to swell up. And this right here, this is a super high-yield point, so make a note of it. Cellular swelling is the very first thing you can actually see under a microscope that tells you a cell is in trouble. And it's not just the cell itself that swells. The mitochondria and the ER inside also puff up like little balloons. But it's not just swelling. We see a bunch of other signs of distress too. The cell membrane starts to form these weird little bulges called blebs. The ribosomes, the cell's protein factories, start popping off the endoplasmic reticulum, which means protein synthesis basically grinds to a halt. The DNA inside the nucleus starts clumping up. You even see these strange, swirly lipid things called myelin figures. Each one of these is a visual clue that the cell's machinery is really starting to sputter. Now, here's a crucial point. Even though we call this reversible, the cell can lose its function almost instantly. Take a heart muscle cell, for example. If you cut off its blood supply, it stops contracting in just 1 or 2 minutes. 1 or 2 minutes. The cell is still alive, yeah, and it could recover, but it's already stopped doing its job. That's how fast this can happen. And that brings us to the most critical part of this story. If that stressor sticks around, the cell gets pushed further and further until it reaches a tipping point. The rubber band is stretched to its absolute limit. This is the point of no return, where bending becomes breaking. So that's the big question, right? What is that critical event? What's the one thing that finally pushes that cell over the edge, from which there's absolutely no coming back? Well, the answer comes down to 2 cellular superstars, the plasma membrane and the mitochondria. See, in reversible injury, the membrane is still holding it together, and the mitochondria, while they might be a little swollen, are still chugging along. But in irreversible injury, you get catastrophic failure of both. The plasma membrane literally breaks down, and the mitochondria get so damaged they can't make energy anymore. Once those 2 things happen, that's it. Game over. And that leads us to the final tragic act, irreversible injury. The cell has been pushed past its breaking point. Its fate is now sealed. Even if we could wave a magic wand and restore its blood supply, it's too late. The death cascade really gets going with that membrane breakdown. The cell's walls are breached, and you get a massive flood of calcium rushing in. Now, calcium inside the cell acts like a wrecking ball. It switches on all these destructive enzymes that just start chewing up everything in sight. And as if that's not bad enough, the cell's own recycling centers, the lysosomes, burst open, releasing their digestive enzymes. The cell literally starts to digest itself from the inside out. It's a process called autolysis. The signs of this fatal stage are crystal clear. First, because the membrane is busted, all the stuff inside the cell starts leaking out into the bloodstream. This is a huge clue for us clinically. Inside the cell, the mitochondria develop these things called amorphous densities, which is just a sign that their energy-producing machinery is toast. And finally, the cell's brain, the nucleus, begins to fall apart. The death of the nucleus happens in 3 phases, and here's the fantastic mnemonic for you, the 3 Ks. First, you get pycnosis, where the nucleus shrinks down and gets super dense. Think of it as being packed. Next is karyorexis, where the nucleus shatters into little pieces. It gets cracked. And finally, karyolysis, where those fragments just dissolve away into nothing. The nucleus gets cleared. So remember, packed, cracked, and cleared. Okay, so that was a lot of cellular drama. Let's pull back now and talk about why any of this matters. How do we use this information in the hospital with real patients? This is where the story of one tiny cell becomes the story of modern medicine. This table is a perfect summary. Let's just quickly run through it. In reversible injury, the membrane is intact, mitochondria are swollen but working, and enzymes are kept inside. The outcome? The cell can recover. In irreversible injury, the membrane is broken, mitochondria are shot, and enzymes leak out. The nucleus undergoes that whole packed, cracked, cleared process, and the outcome is always cell death. And this, right here, is probably the most important clinical concept of this entire explainer. That leakage of stuff out of the broken cell, that's not just a random event. It is the entire basis for many of the blood tests we use every single day to figure out what's wrong with our patients. Let me give you 2 classic examples. A patient has a heart attack. What's happening? Their heart muscle cells are dying. They undergo irreversible injury, they break open, and they release a protein called troponin into the blood. We run a blood test, see high troponin, and boom, we know there's been heart damage. Same thing with hepatitis. Liver cells die, break open, and release their enzymes AST and ALT. When we see those levels spike in the blood, it tells us there's active, irreversible injury happening in the liver. It's that direct. So we've followed our cell all the way to its bitter end. We've seen exactly how it dies, and we've learned how we can detect that death with our lab tests. But that brings up another really interesting question, doesn't it? What happens next? The cell is dead. How does the body clean up the mess? Well, that's a story for our next explainer, where we'll get into necrosis and inflammation. Thanks so much for joining.